understanding and addressing Parkinson's disease pathophysiology in fly model and therapeutic implications to human. In my talk, I try to brief the importance of the animal models with special reference to the Drosophila relating to dopaminergic neurodegeneration, which is the characteristic pathophysiology of the Parkinson's disease, and what are the possibilities by taking advantage of this model system, how one can make efforts to protect the dopaminergic neurons under a disease condition, and how this knowledge generated through these animal models of the Drosophila system, how it is important, how much it is relevant, whether it is really it can be addressed to human pathology, because most of the times, most of the people who are the non-Drosophila biologists, they think that fly is fly, human is human. Yes, fly is definitely the fly, human is human, even microbe is microbe. However, most of the insights relating to multiple molecular pathways, relating to multiple biological processes, the knowledge is generated from the prokaryotic system. So what is happening in the aerobic bacteria or anaerobic organism, the fundamental processes, it may be the respiration or relating processes, the pathways and the molecular players, they remain the same, suggesting the evolution of the life. Okay, fly is a small guy. It has helped the mankind in order to figure out many riddles, which is really the fun. Yeah. So this is, even many times Bharat sir said the serendipitous events. I was just looking into the obituary of Nancy Bonini, who is a towering personality in the neurogenetics using a fly model, who happened to be a postdoc in Seymour Benger's lab at the Caltech. It's a beautiful story. There are many chances. Life is a package of full of these chances, but chances can be amazing, exciting, and refreshing. Here is one serendipitous, one chance event. Seymour Benger, the father of the neurobiology, in specific Drosophila neurobiology, by training he was a physicist. He is a PhD in physics. Then he happened to go to Cold Spring Harbor. Cold Spring Harbor laboratories, they are the Mecca of the molecular biology, and this is way back in 40s, 48, where Benger was working in a bacteriophage laboratory in 1948. So the physicist, by training, a guy is a physicist, and then he tries to understand, by taking advantage of the bacteriophage system, the fundamental processes relating to molecular biology, like cis-trans test, as students we all have studied. Okay, so here, what is that? The Benger, the deletion crosses. So he was trying to show the R2 mutations in a sequence. In simple, the dark and the light, these are the revertants. There are mutations relating to the R2. That's what he has done. It is a pioneering work in the field of molecular biology. So these are the revertants. That means if there is a mutation, it can come back and it can become wild type. This, in simple, we call revertant. So with these elegant pioneering experiments, Benger could used the phase for placing R2 mutations in sequence for which Benger is known. Okay, so the guy who is a physicist by training then goes to Cold Spring Harbor and tries to work with the bacteriophage. So what happened? The chance event, who is a physicist, then phage biologist, then the world famous behavioral geneticist. What is the chance he went there? He was standing in a queue for lunch in Cold Spring Harbor Laboratories. So somebody calls for, like it's a common thing. Nobody will come and ask you, those days there were no screens and all. In 40s we are talking about it. Somebody was just shouting, is there anybody to sign up for the Woods Hole embryology course, which, Drosophila has been, traditionally one of the fantastic model system in order to understand the developmental biological, the processes relating to the developmental biology. Of course, now there is for the developmental biology, Sidney Brenner's great contribution, the worm, it has outsmarted the Drosophila as far as the development is concerned, because the question you ask directly, one can really follow under the microscope the real process of the development, which may not be as good as in case of the Drosophila. So Benger, by chance, he signed up for the developmental biology course. Then he loved the Drosophila. This is in Benger's office in Caltech, where he is looking into the eye of the Drosophila. So then he rest is history. See, people always say that Drosophila is there, right? It may be the Parkinson's disease, maybe the cancers, or other human related disorders. How far the knowledge generated from Drosophila can be applied to the human? This concept I would love to substantiate the argument that the fundamental mechanisms between the Drosophila and the human extremely conserved. Maybe in sometimes there are exceptions. For example, even the alpha-synuclein. Alpha-synuclein, which is involved in the Parkinson's disease, dopaminergic neurodegeneration, the mutants of the alpha-synuclein A30P, A53T have shown the overexpression leading to dopaminergic neurodegeneration in Drosophila, but Drosophila doesn't have the endogenous. There may be certain limitations, the genes which are not endogenous, if they were not to be present in the fly system. So here, way back in 1970s, it's important to see the time scale. The argument, still there is an argument in 2019, 2020. This in 1970, Seymour Benger and his student Ronald Konopka asked whether it is possible to identify genes that control the circadian rhythm in Drosophila. So they generated the mutants. Fly system has been known for, yesterday I was mentioning during the practical time. Drosophila, we are taking advantage of the biochemical methods, but the real power of the Drosophila is in its genetics. You can randomly, like there is a EMS, ethyl methane sulfate, that is one has been, traditionally it has been used in order to generate the mutants. Randomly you just feed the Drosophila, so point mutations will be generated. All over the process is random, and you can screen for the phenotype of one's interest and then can identify the gene, and then the rest of the story follows. This is what traditionally Drosophila biologists have been doing it, but with the advent of the molecular biology, these days there are many techniques, transposons, RNA, of course the latest CRISPR-Cas9. So in 70s, what Benger has done, they want to understand, actually this Ronald Konopka, the student of the Benger, is it possible to understand the circadian rhythms, the genes that are responsible, the genetic basis of the circadian rhythms. So they generated the mutants, and they identified in an unknown gene disrupted in the circadian clock of flies. They named this gene period. It is important to remember. They named this gene period in 1970. Yeah. So the Yoshiki Hotta, he is another postdoc or PhD student in Benger's lab. He is a towering personality in neurogenetics. This guy is from Japan. He was with Benger in Caltech, and the Yoshiki Hotta, he characterized the locomotor rhythms of the normal and period, the gene, the period circadian mutants, they measured with activity monitor. There are multiple ways, as we discussed yesterday. You can monitor the circadian rhythm, locomotion, even the behavior. These days there are assays basing on the sexual activity, the courtship markers. One can characterize, one can quantify even the memory long term and short term, and one also can screen successfully long term and short term mutants. So this gentleman, so what he has done with the activity by monitoring the activity, he characterized the normal Drosophila and the period mutant circadian, and they have shown that, yeah, there is a problem with the circadian rhythms. Maybe after 50 years later, it is the interesting point is, in 2017, there is a Nobel Prize was in physiology of medicine was jointly given to Jeffrey Hall, Michael Rosbash, and Michael Young. What Jeffrey Hall, Michael Rosbash, and Young they have done, they figured out the molecular basis of regulation of the circadian rhythms in Drosophila, the knowledge of which is exactly can be applicable to plants and animals. To cut short the story, what Jeffrey Hall and Rosbash they have shown, see, the idea is like this. The period is the gene product, the period, it controls the circadian rhythms. During night time, it is accumulated in the nucleus and it represses the period activity. That is important. Whereas the fundamental question is, the period is what these two guys have shown, Jeffrey Hall and Rosbash. It is the period which is in cytoplasm, and during the daytime and during the night time, as it approaches, it enters into the nucleus and it suppresses the gene. That is important for the circadian rhythm regulation is concerned. Fine. Then the fundamental question is, if the period is entering into the nucleus during the night time and it is repressing or it is regulating the activity, how this guy, in a time scale or in a circadian rhythm specific fashion, he is entering into the nucleus. Who are his friends? This is where, with a great screen, Michael Young has shown a period. He has a friend called timeless. Timeless is another protein, the period and the timeless, and together timeless is the one that is helping the period to translocate into the nucleoplasm and repress the gene. So the point is, both of them, all the three guys, they shared the Nobel Prize. Whereas the real period locus, it was described by the Seymour Belanger and his student way back in 1970s. It took 50 years to figure out how exactly the process works and how the circadian rhythms are regulated. Yeah. See, now we know that in human or the plant at 12 noon or evening, certain activities are at the high, maybe the night time certain activities are low. Similar thing in the plants, like somebody has shown one particular plant, you take it in light, the leaf behavior and dark, it will be different, but even when it is moved to the light, because of the circadian rhythm regulation, the phenotype would not alter. What is the driving point here? The period and timeless, the association, how they regulate, the fundamental mechanism is worked out in Drosophila. Then they could figure it out, even the call center workers, people who have disturbed sleep patterns, it leads to multiple disorders, in specific multiple neurological disorders. There the regulation of the period and the timeless is exactly the same, the molecular mechanism of the period and timeless between Drosophila, mice, plants and humans, it is exactly the same. Suggesting biological implication is as following. The molecular basis or the mechanistics, the knowledge relating to the mechanism of multiple genes which are conserved, not only the homology and orthology, the biological function remains the same, suggesting in specific there are genes that are involved in the development of the brain and the neurological disorders. Drosophila can be a fantastic system because the major limitation in order to understand the pathophysiology of the any neurological disease, it may be the Alzheimer's, Parkinson's, spinocerebellar ataxia, availability of the tissue sample number one, and availability of the tissue samples with reference to the time point. It is important to understand, you can get the human brain samples, but in order to understand the progression of the pathophysiology, it is important to follow the pathology with reference to the time scale, which is next to impossibility in human. Here comes the model system like the Drosophila. It gives a fantastic opportunity to the human in order to understand pathophysiology of any neurological disease. Not only neurological diseases, our emphasis is neurological diseases because our lab works a little bit relating to the dopaminergic neurodegeneration. Otherwise, many of the molecular mechanisms to figure out Drosophila can be of great help. Yeah, this is what as far as the neurodegeneration is concerned. I briefed this setup once again. You know, Benger, the beauty is, Benger is an amazing guy. That's what, you know, like it is important, the abstractness in science is very important. Maybe about the same thing, if 100 people are asked to tell, 99 people can tell the story. But it is important to drive the point. In order to drive the point, driving a point is a kind of akin, similar to scoring the phenotype, which is relevant to the disease we are trying to understand and address. So, the beauty with the Benger, by being a physicist, you know, traditionally biologists, it would be maybe offending to say storytellers, but we are very descriptive. You know, like we say, you know, fly head is like this, brain is like this, you know, we describe. But it is very important in order to understand, you know, the real things from the apparent things. It is important to split the phenotype. So Benger, he made this was once again he was in Caltech. You know, this is the point I want to say. In most of the time, this can be a fantastic system in Indian universities or anywhere, even sitting in Caltech with maybe, you know, $1 equipment, he can create amazing things, which means our question must be important. The methods, what we use in order to understand and address the question we are asking about, I think, you know, the kind of methods the approaches one adapts, it should be easily doable, which there is every possibility. So the point is, the simplicity of the methodology is, you know, it is very, very essential in order to understand the complicated processes, including the development and pathology relating to the human or the Drosophila brain. So this is the countercurrent system, and over a period of time, first cycle, second cycle, third cycle, fourth cycle, then maybe I think 15 cycles they screened it, and they figured it out, one great mutant which has excited the scientific community for coming maybe 40-50 years, that is drop dead. So essentially the idea is Benger's. So here most of the tens of the Drosophila laboratories, this looks simple, but to score the, you know, finest aspect of the extent of the neurodegeneration, I think it is important to have an insight for the bench worker. What is that the fly labs do, including our laboratory? This is called, we popularly called negative geotaxis assay. We know phototactic, geotactic. So it goes to this guy, Drosophila, if there is a light, he likes the light, he loves the light, so he goes near to it. And similarly, negatively geotactic. So if you put it's a small tube, even the one we use in laboratory, they are the, you know, plastic tubes, but there are, you know, designated these days, there are specific cylinders. it is not important. Even if you have a small glass tube, that is good enough. Only thing is you need to optimize your method according to your situation. The idea is, if the flies, this is the source of the light, as mentioned, the guy moves towards the light. So you put the Drosophila and gently tap it, all the guys will come and switch on the light and guys start moving up. So this simple, I said, the basis is again Benger's brilliant behavioral. It is important to understand the behavior of an organism in order to take advantage of this behavior as a marker to understand and decipher the pathology model of that particular organism for a human disease. So this guy goes, and this is in the dark condition. What we do, you put maybe one here. See, what is the adaptation of the Joes is, here they put so many, maybe 20 or the 25 Drosophila, and they try to see how many people, how many of the Drosophila they cross the specific line, and they count it and they give the ratio. Okay, maybe if you put the 25, 15 by 10, maybe if it is a normal case, 21 by 25 perhaps. If it is a mutant, maybe 20 by 25, 15 by, that is one method. But in our laboratory, what we do, this is the method we have adapted from the Botella, who is a scientist in Stefan's lab in Regensburg University. The method is more sensitive for the guys who are interested in simple. You give every fly 3 chances, because there is every possibility the same person, for multiple reasons, performance can be affected. It's like, so we also give examination for the guy. If I fail in March, maybe the June or August I can take the improvement. Similarly, give the Drosophila 2 improvement tests. First you give it and take a stipulated time, maybe the 10 seconds or 12 seconds according to your situation, and you note what is the distance that particular flight traveled, and give 2 more chances and record it, take the average of it. So for every group, if you have the control or the mutant or some drug rescue or some other thing, drug per se, every group for 25 guys giving 3 chances for every Drosophila, and you can measure the fly speed. The speed of the organism Drosophila is with this negative geotaxis assay, it is a very critical because of the following reason. In order to screen thousands of the mutant lines, in order to understand whether there is any in that particular deletion or in that particular mutant line, is there any possibility of the figuring out the dopaminergic neuroprotective or neurodegenerative phenotype? The primary phenotype should be, the primary screening methodology should be simple and doable. Yes, one can do by the confocal microscopy or the fluorescence microscopy or the HPLC. Yes, it is doable. And even tyrosine hydroxylase with the Western, all this is possible. So if you want to screen 100 lines, it may take 10 years. So the point is, by taking advantage, one can understand the mobility defect is linked to dopaminergic neurodegeneration. But this cannot be an absolute statement, because when the mobility is affected, not only the neuronal system can be affected, but there is every possibility even the muscular system also is affected. So in order to carry forward the story further, the worker, the researcher has to convince himself and look at it carefully. The phenotype one is observing, it is due to neurodegeneration, not due to muscular degeneration, because some of the guys, they also screen muscle degeneration using a kind of similar phenotype in Drosophila system. So this is the system we follow in our laboratory to do multiple experiments. Yeah. So another what they have done, most of the methods from their lab. Another important, very powerful method in order to understand the brain neurodegeneration is plastic sectioning. What they do, that drop dead mutant Hota, so he processed the brain of the fly there is a regular fixation protocols, small fly brain, and you make the sections of it, maybe 10 micrometer, 15 micrometer, according to your the optimized method. You see here, this is the normal eye, ganglia, and this is the brain. When you see the brain of the Drosophila in the wild type fly, it is normal. Normal means there is nothing like, phenotype can be different, but still brain can be normal. So it is important to cross-validate both the ways. But at least structurally, it is normal. In Benja's lab, this is the drop-dead mutant they have characterized. In the drop-dead mutant, in the ganglia and even in the neurophil, the brain region, there are full of holes, suggesting this way they characterized the drop-dead mutant. Of course, it dies young too. The drop-dead mutant, it leads to neurodegeneration specific to the brain. This is how they characterized. And even today, we also, all the fly labs to begin with, that's what other day we were discussing about it, even Melfini's lab, they make the plastic sections in order to understand the dopaminergic neurodegeneration. But now, of course, it would be nice if you can see the whole brain rather than the sections, because there is every possibility, particularly because here it is very, very evident. Whereas sometimes if it is only a specific cell type like the dopaminergic neurons, there is always a wishful thinking. So the better way, but we are talking about the 70s and the 80s, today even 2002 they adopted the same similar method, but now most of the laboratories, whoever has got the little bit infrastructure, they look into the whole brain rather than looking into the sections of the brain. Yeah, so Benger got Albany, they say it is equivalent to the Nobel. So he says, experience thus far with the fly as a model system for unraveling the path from the gene to behavior is encouraging. In any case, it's fun. So one thing before I want to say is, even in those days, Benger, he gene expression profiles in the Drosophila aging brain, he worked on it, and with the RNA in situs, he has shown the expression profiles between the Drosophila brain and the human brain, they are pretty similar. This is in maybe 2002 paper of the Seymour Benger. So whole world was excited because of this paper. They thought it's an amazing thing. What is happening in the fly brain is pretty similar to the mechanistics of the human brain. Okay. So dopamine, there are many neurotransmitters. Neurotransmitters, we all know they are chemicals. As somebody said, life is nothing but half a dozen chemicals. So dopamine is a neurotransmitter. It is released by the brain that plays number of roles in humans and other animals, including the Drosophila. So any kind of novelty or excitement drives up dopamine in the brain, and dopamine is associated with romantic love. So somebody said, Vaughan Bell is a great artist. So once he called, just I was looking what people thought about the dopamine. He said the dopamine is Kim Kardashian of all molecules. So just I was curious, who is this Kim Kardashian? Of course we heard about her. So why he said it. Then when I googled it, this is Kim Kardashian. Now we know that yes, it is the dopamine is definitely the Kim Kardashian of all the molecules, suggesting dopamine is an amazing thing. Virtually, it rocks many of the life processes, including movement, memory, the pleasurable rewards like if you take a good food, when you are excited, and sexual activity, courtship behavior, and several drugs. And even dopamine is behind the attention. Attention deficit syndrome is also due to the altered levels of the neurotransmitter dopamine in brain, and inhibition of the prolactin production, sleep, mood, importantly the learning, and even the cocaine and amphetamines inhibit the reuptake of the dopamine. The perturbance in the dopamine reuptake mechanism is also leads to multiple neurological disorders. So this one was first recognized by Carlson, and Carlson was awarded 2000 Nobel Prize in Physiology of Medicine for his discovery relating to the dopamine. L-dopa is a precursor of the dopamine, which was effective treatment for the symptoms of the Parkinson's. And there is also a group of neurobiologists, they believe dopamine also involved in the schizophrenia, and it is sensitive to the aging process, varies for the different transmitters and different regions of the brain. Dopamine neurons are more age sensitive, suggesting it is not that only neurodegenerative diseases or some pathology conditions there is a loss of dopaminergic neurodegeneration. With the age, there is a natural degeneration of the dopaminergic neurons, which leads to multiple processes normally, maybe the loss of memory, all the things related to the natural degenerative processes associated with the aging process. So as we know today, aging is a systemic, but even in the systemic process, certain cell types are more sensitive in the brain. One of the critical cell type is dopaminergic neurons. Therefore, with reference to the quality of the life in the aging population, the dopaminergic neuroprotective strategies are important and critical in order to promote the healthy aging process. So the Parkinson's, it is about 200 years ago, English physician James Parkinson, he has described it. It is after the Alzheimer's, it is next common neurodegenerative disease. It has got the sporadic and genetic components, as is the case in case of the cancers. 95% of the Parkinson's disease patients, it is sporadic. But with the advent of the alpha-synuclein is the first gene that is implicated in the Parkinson's disease. So today we know about alpha-synuclein, DJ1, UZHL1 and Parkin. There are about 12 genes and 10 different loci. They are implicated in the degeneration of the dopaminergic neurons. So which means the environmental factors are more critical for the health of the neuronal protection. It is also epidemiological studies, they show that it is less prevalent in India and some Asian countries, suggesting perhaps it may be due to their food habits. Here comes the importance of the nutraceuticals such as curcumin. And it is also epidemiological studies, they also show that the prevalence rates in men are slightly higher than in women. So this is attributed to the neuroprotective efficacy of the estrogen. Okay, clinical features, we know the resting tremor, there is a slowness of the movement, postural instability, dementia. The loss of intellectual faculties is the critical pathological feature, and importantly, loss of dopaminergic neurons in the substantia nigra pars compacta. There may be many regions, it's not that dopaminergic neurons are exclusively in the human brain at one place, but even within the same cell type, there is a regional specification is there. The loss of dopaminergic neurons in the substantia nigra pars compacta region of the human brain is one of the critical pathophysiological feature of the Parkinson's. Lewy bodies are intracytoplasmic proteinaceous inclusions. These are, the idea is, one of the popular ideas is, if you are going on a highway, if there is an accident, there will be blockage of the vehicular movement. So the intracytoplasmic proteinaceous movement, they may part of the cargo movement, which may be one of the critical factors for the neurodegeneration of the dopaminergic neurons. Yeah. So I think enough we talked about the Drosophila. The point is, as far as the disease, study of the disease pathology is concerned, progression is concerned, certain important things, the short life cycle. Drosophila has got at the 20 to 23 degrees centigrade, and if you keep an incubator, it may be the 10 days, first instar, second instar, third instar, pupa and the adult. Which means, if you want to study multiple, if you want to score multiple generations, within a short span one can score, which is a very critical thing, particularly in the genetics experiments. For example, the idea is, in order to change the background of the genetic background, when you make a crosses, short life cycle of an organism, it is really useful. Imagine if even if it is a mice, the mice life cycle, mice longevity is more, when compared to the Drosophila, it will be more hectic, whereas because of the 10-day life cycle, Drosophila is very handy in order to score the generations. Great genetics, of course, again and again it is because, see, any organism, anybody can, any other fly also one can use it. It is not that the Drosophila, but there is a tradition, availability of the mutants. For the Drosophila, you have the EMS generated mutants, you have the US Galfor system, which is an amazing system for the loss of function and gain of function analysis. US Galfor. Galfor is a transcription factor. If you downstream of the US, if you can screen any gene in a tissue specific fashion, it is possible either to overexpress or knock down any gene. That means, in order to understand the loss of function, in order to understand the function of a gene, the loss of function, gain of function analysis, the required tools are available in Drosophila, which makes the effort of a researcher quite handy and doable. Then deletion lines also there these days, RNA lines also there, so that one can knock down the genes. Then importantly, it is not that there are methods I would work with any organism. There is a conserved genes and molecular mechanisms. Whatever the most of the disease causing, 70% of the disease causing genes, the structure is conserved, and it is not only structure is conserved, orthologous, even the function is conserved. One best example, as I have shown before, the period and timeless. In fact, period and timeless, first they were discovered in Drosophila, then went to the human. Same thing with the gene involved in the hippo. There are many neurological diseases, the genes involved in the mitochondrial biogenesis. They were first identified in Drosophila, then people went to the disease cell lines, mice, of course, and to the humans. Amenable behavioral analysis, it looks simple, but it is an amazing thing, as from the Benger's time, negative geotox assay, negative geotoxis assay, and countercurrent mechanism, countercurrent assay. So to assay the disease phenotype, that is the critical. So fly can be very powerful for that. Yeah. So what is that the idea is, what is critical in order to understand, one needs a model. So we developed a model. What is known is, epidemiological studies show that there is a correlation between exposure to the neurotoxicants when someone is younger and onset of the neurodegeneration at later phase of the lifespan. So what they have done, see, it is very important, epidemiological studies, these clinicians, as a country, we lack this tradition. If somebody has a Parkinson's disease or any particular kind of cancer, they will go to a clinician. It is important to, if you see the end slide, there is a death of dopaminergic neurons or some kind of cancer, that is the end slide. But there is a journey, like how human has the journey, cell has its own journey. What happened in the process of it's a travel. So people, they figured it out, okay, what you were doing when you were young, East Asian countries, most of the East Asian countries, these epidemiological studies were performed. What happened, these are basically agrarian economies. Agriculture is the primary thing for the people, and rice is one of the critical economic crops. So most of these guys, what the clinician's observation is as following. Many people, they started developing the PD. Until that time everybody knows it is a sporadic, but when you know that sporadic means what? Sporadic means cause is not known. When the cause is not known, even if it may be a cancer if it is a sporadic, and a neurodegeneration is sporadic, if Alzheimer's or PD or something else if it is sporadic, but where is the starting point in order to understand the disease? So epidemiological studies are very, very important. So what is known is these clinicians, they worked out, and the guys who are the sufferers of the Parkinson's disease, in whom the PD is setting in, maybe when they are 20s and 30s, they worked in the agricultural field. Those days in agricultural field, when they try to dig the story deeper and deeper, they got to know that they used to spray the neurotoxicants like the paraquat as a pest control. So they thought perhaps exposure to the certain neurotoxicants where during the early phase of the lifespan, perhaps one of the possible reasons for the onset of the dopaminergic neurodegeneration when someone is at the later phase. That is a frame shift observation in understanding the pathophysiology of the Parkinson's disease. It looks simple, but it is the critical observation. So similarly, when 95% of the PD cases are sporadic, perhaps one of the critical things that is responsible for this degeneration can be exposure to the neurotoxicant. It need not be the agricultural field, it can be an industry, or it can be something else too. And okay, so here what we have done, the Drosophila, in order to develop a model, what we have done is, to an array of these neurotoxicant concentrations we have exposed it, and then it shows the concentration and time dependent mortality of the fly when it is exposed to the neurotoxicant paraquat. But what is the essential difference between some other studies and our studies is, at the time point which we are conducting the experiments and we are showing the rescue phenotypes, there is no mortality, because we are of the opinion that the death is systemic. If you expose to 30 millimolar, 20 millimolar paraquat or any neurotoxicant for 3 days, 4 days, definitely you will see phenotype, and guy also will be dying. So it is important to understand the pathology linked to a specific specific cell type in the brain. The story, it is important, it is not linked to a systemic event which is death. So that care we have taken when we set up the model, actually eventually subsequently this understanding way back in maybe in 2012 or 13, it is really helping us in order to figure out the genetic or the molecular mechanism, because multiple pathways which may not have linked with the neuroprotection or degeneration, they are upregulated or downregulated in if any particular model organism, or including the human, is exposed to neurotoxicant which has got nothing to do with the neuroprotection or neurodegeneration. It is just a systemic influence. So there is every possibility, 90% of the targets what we are seeing, it may not be linked with the dopaminergic neurodegeneration. This is the important point. I think we went slow. It doesn't matter how slow we are. Sometimes it is so when you really want to link the story to the brain, maybe this insight will be useful. This is of our opinion, and it is working for us. Yeah, so less said about the curcumin is better. Curcumin is anti-inflammatory, antioxidant, and more importantly, in our tradition for the past 8,000 years or 6,000 years, we have been taking, as Professor Bharat said rightly, NIH principle is bench to bedside, because they lack the tradition, but our tradition, we know so much about so many good guys in nature. So it is important to modify the protocols according to our local situations. So it is not that the NIH motto is the bench to bedside, but it is reverse. So the simplest thing is, somebody went to Japan, and in some particular island people are living long. People are living long. It is, if you want to figure it out, it may take 100 years, still you may end up with the dead. Point is, it is important to see what exactly is happening to the people. So if some particular population is living long, so what they figured it out, this green tea, all multiple kinds of this mo tea, something in Japan there is a specific tea. It starts with M, I'm sorry I failed to get it, mocha tea, mocha. So they figured it out, maybe the very, very low concentration, the chronic consumption of this, it extends the lifespan. Now people have shown even in the animal models, it extends the lifespan. That is also antioxidant, anti-inflammatory, that is a different story. It is very unlikely all the antioxidants, all the anti-inflammatories, they can protect the dopaminergic neurons, they can address the, they can address the Alzheimer's, they can extend the lifespan. Definitely there must be molecular mechanistics may vary, but by looking into the food habits of the people, it is possible even without holding the test tube, which may work, which may not work. So that is a very important thing. So curcumin, I think we know we don't need any introduction. We have been eating this guy and we are fine. That's what, even if when we go to a party, if some doubt is there, look around, if everybody is taking, we can take it just like that. It is as simple as that. So people have been taking in curcumin. So we know curcumin is a good guy. Yeah, there are many products in the market. Okay, but having said that, there are two things critical in biology or the life system: the path and the dose. What is a medicine or what is a poison? The medicine can be poison and vice versa, depending upon the path and dose. What is path? Path is you take water, no problem. But if you just clip it and if water goes to lungs, it is lethal. Same thing with the dose. Even salt can kill. So dose is the dose and the path, however good they are, it is they are the critical, because cell works with the optimum. Everything cell wants optimum. But what is important is curcumin also toxic in a dose dependent fashion. So actually, somebody, reviewer said, cut off all those concentrations. We have done maybe 20 different concentrations in an elaborated way. Idea is how we can identify the genetic targets. So once again, the concentrations, what we have taken, the curcumin, whether it is a chronic treatment or for the short-term treatment, all are at that particular time point, it is not affecting their longevity or any other phenotype. So what is important is when we try to understand the late onset neurological diseases, Parkinson's and Alzheimer's, they are the late onset neurodegenerative diseases. What is known to the biologists is, in any organism, it may be the Drosophila or the mice or the human, there is a health, transition and a senescent. Most of the late onset neurological diseases, they onset during the transition phase. What is transition? It is around 50 days, which is equivalent to 50 years of the human, considering if the Drosophila lifespan is, let us say, maybe 100 or 100 days, human considering 100. Fletcher has shown the gene regulation profiles is 27 to 30%. They vary among 3 phases of the lifespan, this health, transition and senescent. So what is the driving point? The driving point is, it is important to, when we screen a drug, it is important to understand or the figure out if it is a genotropic drug, as it works through the regulating the activity of the genes. It is important to take care at which point the disease sets in. At the same point, important to understand the efficacy of the protective molecule. So what we have done, most of the laboratories, in order to understand the curcumin or any other molecules dopaminergic neuroprotective efficacy, they have taken the 4-day or 5-day Drosophila. It's like taking a 5 or 6-year-old kid and screening for the neuroprotection. So there are many molecules, anything and everything, they have shown that it is conferring the dopaminergic neuroprotection. So once again, this is the basis for this is, to cut short the story, you expose to the neurotoxicant and you also expose in association with the protective agent, maybe the curcumin, and perform the climbing essay in order to make the preliminary screening. Here we have othered to begin with the co-treatment, two regimes, co-treatment and pre-treatment. It is also important when we say something is neuroprotective, it is necessary to figure out the physical sequestration process. In order to rule out that possibility, we also have followed first to feed the guy, whether it is a prophylactic, if you adhere to it, will it work, will it protect the dopaminergic neuron? What we have observed in the laboratory is it is working both the ways, suggesting the dopaminergic neuroprotective efficacy. What you observe in the fly model of the Parkinson's disease, it is not due to sequestration of the neurotoxin, but it is due to some other mechanism, genetic mechanism, with the help of which it is protecting the dopaminergic neuron, suggesting this knowledge will be of the great help in order to in order to apply for any other model system, including the human. Yeah. So yes, initially we have anybody would screen it in a simple way, but what is critical for the PD pathology? The death of the dopaminergic neurons in the substantia nigra in case of the human patients. Drosophila 2 has got the dopaminergic neuronal clusters, multiple clusters in its brain. This is a kind of, it's a cartoon of the Drosophila brain showing multiple clusters of dopaminergic neurons. But are these the only dopaminergic neuronal clusters in the fly brain? Answer is no. There are also other dopaminergic neuronal clusters. Then what are these? These are the quantifiable dopaminergic neuronal clusters in the Drosophila brain, and we quantified it. And this is not the confocal. With a lot of effort, we developed a novel and sensitive method in order to understand the dopaminergic neuronal dysfunction. What is the story? Traditionally, for the past 20 years, many of the fly laboratories, including the Melfeni's lab at the Harvard, they make the plastic sections and they count the neurons. This is the critical phenotype in order to figure it out whether the neuron is protected or not. But there are multiple workers, they have shown there is no loss of dopaminergic neurons in the fly brain, whether it is alpha-synuclein model or Parkin model or DJ1 model or sporadic model, which has opened the Pandora's box. What does it mean? It means, in simple, really it is not a model. So then people started looking into the whole brain. Some people, they have separated the dopaminergic neuron through the flow cytometry, and they try to quantify the signal intensity. In the Stephan's lab in Regensburg, there is a smart Spanish-born German scientist, Botella. He developed a novel method with the help of which he quantified the neuronal dysfunction. But we don't have access to the confocal microscopy. This is a fluorescent microscope. We learned that with the help of that method, we modified it, and we developed another method, a parallel method, in order to understand the dopaminergic neurodegeneration. The driving point is as following. See, all these, Paul, that is not important, 1, 2, 3, 4, 5, these are the clusters in the dopaminergic neurons. Looking into the whole brain, in 3D axis, we have counted, all characterized with the number of the neurons at every level relating to relating to multiple clusters. And when we quantified it, there is no neuronal number loss, suggesting confirming the observations made starting from 2002. There is no neuronal number. That is what exactly the Botella has been pushing. He pushed it. People, because already tens of papers are published. It was very difficult. End of the day, he could successfully publish it. He started with the Nature, then he ended up in the Journal of Neurochemistry, and he has shown there is no neuronal loss. Here we see that there is no neuronal loss. However, what is interesting point, once again for the non-Drosophila workers, here the red dots, what you are seeing, they are the dopaminergic neurons. How we detect the dopaminergic neurons? In order to detect out of the million cells, 150 cells or 200 cells, if you want to, you need a marker. What is the marker here? Marker is tyrosine hydroxylase is the rate limiting step, rate limiting enzyme in the synthesis of the dopamine. This is the primary antibody raised against the tyrosine hydroxylase. So what is important point is this tyrosine hydroxylase antibody, it is raised against the human dopaminergic neuron, human tyrosine hydroxylase. Human tyrosine hydroxylase raised primary antibody, it is interacting, it is cross-reacting with Drosophila dopaminergic neuron, suggesting the dopamine anabolic and catabolic, particularly at least anabolic process, it is conserved between the Drosophila and human. So human tyrosine hydroxylase antibodies raised against the human tyrosine hydroxylase, then secondary fluorescently labeled antibody, then see under all the sections, minimum 5 brains for the group, all the sections make it, merge it, and quantify the pixel intensity that is emanating from the secondary fluorescently labeled antibody, the intensity of which is directly proportional to the extent of the tyrosine hydroxylase that is synthesized, which provides a critical insight in order to understand the health of the dopaminergic neuron. That is the idea. So when we try to do this, here as you see the same clusters, 1 is 1, 5 is 5. Here there is no difference in the number, whereas the pixel intensity is less when on the exposure to the neurotoxicant, whereas the disease phenotype relating to the dopaminergic neuron is significantly rescued. Some cases partial rescue. It is rescuing, replenishing the tyrosine hydroxylase level, suggesting the curcumin is working and the method is working. So this is this part of the data we are yet to push it. We are excited about it because it is working and it is optimized, and the fine level of the neuronal dysfunction can be quantified using this method. The story is, during the health phase, what I have described before, it is rescuing not only the mobility defects and also the neurotransmitter, the dopamine level is replenished, suggesting curcumin has the dopaminergic neuroprotective efficacy during adult health phase of Drosophila melanogaster. But flip side is, health phase is not the phase where this late onset neurodegenerative disease sets in. So what is happening during the transition phase? This is the story during the transition phase. Here when we say the transition phase, approximately Drosophila is around 50 to 55 years, which is equivalent to the human 50. It's a thorough experiment with a lot of effort. Our boys, when they characterized it, yes, there is no loss of the dopaminergic neurons as far as the number is concerned. Yes, there is a pixel intensity reduction, suggesting the reduced levels of the tyrosine hydroxylase, leading to the reduction in the level of the dopamine, which is not rescued with the curcumin, suggesting curcumin, it is corroborating our earlier results showing the negative geotaxis assay mediated mobility defects are not rescued by nutraceutical curcumin. It is rescued during the health phase, whereas it is not rescued during the transition phase, suggesting the limitation as far as this story is concerned, the limitation during the transition phase. We don't say it is not working, guy is good, fantastic, he is working, he is capable enough, but for some reason, when you age, he is not, his ability is not sufficient enough to protect the critical cells in the brain, which are dopaminergic neurons. Yeah, this is okay. This is a cumulative data. We thought, because we want to understand, as is the case of the human, among the subcell types, among the cell types, is there any cell type specificity? If there is any cell, if there is a cell type specificity of the neurodegeneration, is the degeneration process is the same across all the subgroups of the dopaminergic neurons, or it is different? We performed the first experiment, and to make it more clear, when we merge it, whatever the phenotypes we have described, what I mentioned before, it is exactly the same. Even the overall, there is no neuronal number degeneration, pixel intensity reduction profiles, they remain the same both during the health and transition phase, suggesting the hypothesis we are trying to push, it is matching at the level of the dopaminergic neurons and mobility defects too. Okay. Yeah. So what we did, yet when we did it in 4 years before, we did not have the HPLC system, so we got it done only dopamine, but meanwhile in 15 we got DBTs, thanks to the DBT, Unit of Excellence in Biotechnology. They supported us very liberally, and once again we thank to DBT. We got many, including the fluorescence system. Here what we did, we optimized in our laboratory the metabolites of the dopamine, which is HVA and DOPAC. So this is critical because it is easy to tell the story, dopamine is down, but it is important to understand what exactly is happening, whether it may be due to the upstream tyrosine hydroxylase, or it is every possibility the upstream during the catabolism of the dopamine, because in humans the suppressors of the two critical enzymes COMT, catechol-O-methyl transferase, and MOA, monoamine oxidase suppressors are the critical therapeutic agents in order to protect the dopaminergic neurodegeneration. So therefore, though in animal models it is stopped by looking into the dopamine levels, it is important to understand what exactly is happening, the downstream process, which would provide the comprehensive picture of the activity or inactivity of any nutraceutical, or even in any mutant system, the function of any novel gene. I think we strongly feel, even logically, it is important to understand. So successfully, with a lot of effort for about 2 years or so, once again we optimized all these players. We also optimized the 5-hydroxytryptamine serotonin, which also directly or indirectly it has a role. If we want to see circadian rhythms, or somebody wants to understand the circadian rhythm, that is a fantastic player. As far as if you see the timeless or period, there is a perturbations in the serotonin and related neurotransmitters. So this gives a fantastic greater opportunity to understand further, to dig the story in a sensitive fashion. Okay, so this is the level of the dopamine is down and replenishing, and it is not being happen during the transition. Yes, then shall we break for 10 minutes and once again we can continue? It is 1 hour, so or we can continue after 10. We can have some tea, and is it okay? For 10 minutes quick break we can take it, and once again we can do that, because it is up to 12:30 only today, sir. Okay, so up to this is the part. So next what we want to do, we want to understand, because slowly the excitement in multiple neurological diseases, including the cancer, is emerging to mitochondria, the role of the mitochondria. Mitochondria is a very critical. He came to us as a guest, but when the guest is very important, cell did not leave him. So this guy is very critical for the health of for prosperity of the house, which is cell. So we want to see the mitochondrial mediated dopaminergic neurodegeneration. Can it be prevented? We continued the story that will further will go ahead after a while. Okay, thank you.